With that in mind, I imagine some of you may already have done functional connectivity projects or sort of be familiar with some of these tools. And so hopefully there will be sort of obvious points that I've sort of added in about references or other higher level questions that you may want to ask. If so, even if I'm not talking about it, feel free to just interrupt and let me know if you have questions on those points, and we can dig in a little bit more deeply as the conversation sort of takes us. And of course, also, if you have basic questions and just things that aren't clear, feel free to let me know, and I'm happy to go over that. Okay, so I wanted to start off the talk actually, just in case we're running late at the end, with the acknowledgements. So I make sure to acknowledge all of my collaborators that I've worked with on these various projects, and in particular my current lab that's really taken the lead on doing a lot of the functional connectivity work that we do. And I also wanted to put up this slide to put up my contact information. You can contact me through email or Twitter or whatnot in case you have questions that you don't manage to ask during this session, and you can get in touch afterwards. And I do also want to make a little bit of a plug, which just to say that I will probably have new RA and postdoc positions opening soon in the lab. So if you are in this position or you know someone who's in this position, do feel free to sort of contact me for more information. I'd be really interested in hearing about your background. Okay, so I'm going to start off the talk today by sort of emphasizing that where these functional connectivity methods are placed within different viewpoints of how we might study the brain. And so a lot of the work that's been done in neuroscience has been focused on trying to understand the specialized processing that goes on in different regions. So how a particular region or a particular neuron may even encode very specific properties, be tuned to particular properties, say, of a visual stimulus or where attention is placed in space. And so in contrast to this localized processing perspective, another perspective on brain function emphasizes instead the interactions that are occurring between different brain regions, so a more distributed processing perspective. And of course, we might logically think that both of these must work together for brain function, but with functional connectivity approaches, we're really focusing, instead of like more basic evoked activation style approaches, we're focusing more here now on understanding the coordinated interactions that are happening between different brain regions. And so I'll sort of outline the talk today. In the first section, I'm going to tell you a little bit of very general background on functional connectivity and how these measurements work. Then I'll dive into a few recent projects that we've done just to give you some sense of why you might make these measurements and what they can tell us about the brain. And then I'll tell you about the tutorial that I've designed as an intro for functional connectivity. Alrighty. Oh, and I wanted to, you probably are pros at this already at this point in the week, but I'll just sort of point out here that if you wanted to already open up and start thinking about this, you can open up the tutorial through the Jupyter Hub that Ariel has put together. You can also, if you're doing this later, want to interact with it, you can download it either from my website or the NeuroHackademy website directly. And there are some pieces of the tutorial that use a free toolbox called Gephi for graph visualization. You don't need to have it. You can sort of engage with other toolboxes for graph visualization. But if you wanted to follow along with all the elements I've listed there, then you might want to download that. Okay, so I'm going to go ahead and get started. And so I want to start off by pointing out that for the experiments I'm talking about today, we use functional magnetic resonance imaging to measure these functional networks through the blood oxygen level dependent signal, which tends to increase with increases in neural activity. But and so while I think fMRI is a really great tool for measuring functional networks, and sort of I'll make the plug that it's a great tool because it gives us this really wide field of view. We can measure more or less the whole brain at once, more or less approximately simultaneously, which is a really great way, great characteristics for measuring large scale networks across the whole brain. But of course, there may be advantages to using other tools, and these sort of techniques have been used with a variety of different measurement modalities, EEG and MEG, or more invasive signals from ECOG, optical signals, or even measurements from multi-unit electrode recordings or the like. And so a lot of what I'll be talking about, while it's contextualized within fMRI sphere and fMRI measurements, can be ported relatively easily, I think, to these other techniques, although you may have sort of different aspects of the signal to play with within each of them. And so when I say functional connectivity, I'm just going to start off by, or functional networks, what I am referring to with this term is fairly simply just a measure of the statistical association between the activity patterns of different brain regions. And most often it's just correlation-based measurements. So we take the activity patterns between two regions and we might correlate them to say whether or not they're quote unquote functionally connected. Of course, correlation is not the only measure that you could take of statistical association, and you could use coherence-based measures, you could use PCA, ICA, and so forth. And all of these measures at some point or other have been used in order to measure functional connectivity. I'll focus most on correlation, but we can sort of also talk a little bit about these other techniques if you're interested. So to make this a little bit more concrete, when we say functional connectivity, typically what you end up doing is you take the average time course of activity from different regions of the brain, for example, left and right motor cortex for these, say, spheres that I have plotted on here. And you might measure that bold activity over time. And you could see that these two time series look very similar to one another, and indeed, they're very highly correlated with one another. Another region that's part of a different functional system may instead show a very distinct pattern with much lower correlation. So left visual cortex is much less correlated with motor cortex than the motor regions are to each other. So what was surprising when this was first observed is that this is true not just when I ask someone to engage in a motor task, but even when someone is sitting as still as possible in the scanner at rest, their left and right motor cortex activity patterns are correlated with one another. And indeed, this has been shown to be true even, say, during sleep or very different other states. And in fact, the patterns of correlations in the spontaneous activity mimics the patterns of evoked activations that you might see in a task-related context, for example, in a finger movement task. And this extends beyond just cortex. I showed you left and right motor cortex here, but in fact, a single motor cortical region might be correlated with motor-related regions also in the subcortex and cerebellum and so forth. And so when we make functional connectivity measurements, you can make them not just for a handful of regions, like I showed you on the previous slide, but many different regions spread throughout the brain at once. And one way that you can visualize all of these correlations at a single moment in time is through a correlation matrix, as shown here. And so in this correlation matrix, any given cell just represents a correlation between a pair of those regions' time series, as I showed you on the previous slide. Now, one thing that's important to keep in mind as you make correlation matrices is that order matters quite a lot. So here I've ordered the regions randomly, and what you can't really get much of a sense from this correlation matrix of what's being shown. It just looks like kind of this noisy, staticky diagram. But if I reorder the regions such that I placed ones that are part of the same functional system close to one another, you see this really beautiful structure emerge. It's often called the block diagonal structure within network world, where you can see that there are high correlations in these blocks along the diagonal, that is within a given system, and relatively lower correlations in the off diagonals, that is between different systems. And so because of this nice organization, we can very effectively use different community detection algorithms to identify systems within this network, this large scale brain structure. And I'll talk a little bit more detail later on during the tutorial section about different community detection algorithms you could use, but suffice it to say there are a number of different ones that have been employed, and you might get something that looks like this. So I've drawn out with different red lines the different subnetworks or systems of the human brain, as shown here. I've put labels on them. You could also plot them onto the brain, as shown here, and paint them in different colors. So this is in some ways very exciting, right? This is a technique that uses data-driven methods based on spontaneous brain signals to map in a lot of detail the functional systems of the human brain. I'll sort of pause here to put in a few notes of caution on these methods as well, that again, we can return to, or sort of if anyone has questions right now, we can stop and talk about, that should be kept in mind as you're doing functional connectivity-based methods. And so it starts with this idea that unlike task fMRI, there's no external imposed perturbation. We're just taking correlations in the signals between different brain regions, which means that these measurements can be really susceptible to different forms of noise or artifacts. And so let me walk you through some of the most common types that you might want to be aware of as you're making functional connectivity measurements. So one is just straight up sampling variability. And so by sampling variability, I'm just referring to the variability that you probably learned about in one of your first statistics classes. That is that your particular sample is just an estimate of a statistic from the full population, and it has a certain amount of variability or error associated with it. And like with other statistics, this error tends to go down with increasing samples from the population. And so the same property has been shown to be true with functional connectivity estimates, where if you take single moments in time, or these are different sessions, you might notice that there are fluctuations across them. But it's been shown that a lot of these apparent fluctuations could be attributed to just sampling variability, the amount of time that you have from a particular person, and that the amount of variability tends to decrease as you collect more and more data from a given person. So data quantity is very important for making good functional connectivity measurements. In addition to this sort of more random form of noise, there are also various forms of biased noise that can really influence your signal. Many of you are probably aware of motion as a major impact on fMRI data in general. Functional connectivity in particular, even very small amounts of small movements can really influence functional connectivity measurements. And I'm putting up here a few references of papers that I think have done a really nice job of going through various different denoising methods out there that have been proposed, and sort of looking at the impact of these different pre-processing strategies on removing the biases that come from motion. More recently, it's also folks have become aware that respiration is also a major issue, and there are different strategies that you might use to address different respiratory artifacts as well. In addition to motion and respiration, so these more basic like physiological effects, there may be also other effects such as arousal related effects that you maybe want to be aware of in functional connectivity data. And I put this with a question mark because perhaps this is actually what you're intending to measure, in which case this is not obviously noise that you want to remove. But in other situations, you may be interested in something stable, or you may be interested in a different type of state. And so you wouldn't want to be confounded by arousal related artifacts. And Talyazuki and colleagues, whose figure I have shown here, have done a really nice job of sort of mapping out the changes in functional connectivity that can happen as someone descends into deeper and deeper sleep, showing that functional connectivity is influenced by sleep-related changes, and that we should be cautious about this when we make our measurements, especially because many rescue studies are done with eyes closed fixation. And so, many of us may experience that when our eyes are closed sitting in the scanner at the end of a long day, you may fall asleep. And so you may want to be careful in interpreting the results if people can fall asleep inside the scanner. All right, with all that in mind, though, I will say that functional connectivity methods, so despite these concerns and their lack of constraints, these methods have produced really robust pictures of the typical network organization of the human brain. And so here I'm showing you the results from two different studies. This one was collected at WashU on 120 individuals during resting state. And this one was collected at Harvard, I think on about 1,000 individuals also during resting state. So although these were collected on two different samples, two different scanners, and they actually use very different clustering techniques for these two approaches, you can see there's a lot of similarity in where the typical networks that have been defined across these different methods. And in fact, some of the differences that are present across here are actually have been shown to be just a matter of resolution. So you might break a network apart into two in one case and not in the other. But overall, the group average structure of these networks seems to be really robust. In some of the work I've done, as well as in colleagues' work, we've also shown that you can extend these measurements to produce these techniques to produce very reliable measurements now at the level of individual participants as well. And so I think this is quite exciting for our ability to, again, map functional network organization, both at the group level as well as individual level. And so what can you do with this information? So you can start off by asking questions about the spatial layout of these networks and use that in various ways. But one nice application you can also use these measurements for is from thinking about individual regions or pairs of regions to understanding these interactions in a large scale systems perspective, and sort of adopting tools from graph theory or complex systems world that have been used to study many other types of complex systems like social networks, air transportation networks, and so forth. And so in this framework, I've just sort of replotted the same data, but now here individual brain regions are shown as nodes in this plot, and high functional correlations between these regions are shown as edges in the plot. And so we can now go one step further and take away the sort of anatomical layout of this network and instead use a layout such as this one, where we've now moved the nodes so that we position them where we have nodes that have stronger or more edges between them closer together. And so this is sometimes called a force layout or a spring embedded layout. And so why is this valuable? Why would we want to do this? Well, when we start to look at the brain in this with this sort of graph, certain properties about the system become more apparent than were obvious in the anatomical layout. For example, you can try to identify properties of the network as a whole. So for example, you might notice the clustering of these systems within them. This is the default system, visual system, and so forth. They seem to separate from one another and be clustered close together with each other. And you might also notice properties of individual nodes within this layout. So for example, you might notice that some nodes seem to lie kind of at the center of this graph in between different networks. And these are nodes that we might call hubs, and something that my lab has been really interested in studying. So each of these steps along the way allows us to see new properties, basically, of these functional network interactions. So I'll talk a little bit more about these approaches and measures in the tutorial at the end, but I'll just pause here for a second to see if there are any questions off the bat about this brief intro. Go ahead, Priya.
I was just wondering if you could say a little bit about coherence, because I've heard of, well, I've even used correlation, and I understand ICA and PCA, but when you're talking about coherence, what are you measuring?
Yeah, so that's a great question. So coherence, you can think of as kind of like correlation in the frequency domain. So you move your signals to the frequency domain, you look at the power spectrum, and you look at signals that have similar, within a certain, say, frequency band, have similar levels of fluctuations. And the value of coherence, I think, in functional connectivity world, and Ariel can also step in since he's done quite a lot of work on this, is that not only can you separate things based on different frequencies, which can be quite nice, obviously, from a neural perspective, but can also be valuable for separating out magnitude effects from phase differences. So you can get phase-based measurements of the lag between different signals, which you can do with correlation as well with lag-based statistics, but it falls out quite nicely from coherence-based measures.
Thanks.
Yeah. Go ahead, Harvey.
Hi, I'm not sure if you're getting to this later, but since you have the force graph here, I previously worked on some projects that involved graph theory and especially projecting and using Gephi. And at the time, something that I had to think about was which force-directed algorithm to use, because there are a bunch. I think we ended up using Yifan Hu for most of our Gephi presentations. And even within single algorithms, there might be parameters to tune, and it's not deterministic even at a single set of parameters. So how do you kind of work out on maybe what type of force-directed algorithm that might be optimal to present? And then on top of that, I don't know whether or not that consistency comes into a picture for you.
Yeah, I think that's a great question and a difficult question. We've also sort of struggled with this, and we played around with different ones. Honestly, what we've tended to look for is consistency. We use this, I think it's a really useful way of thinking about and looking at your data, but it's not necessarily a super useful way of quantifying your data ultimately, because it can vary across these different layouts, also across thresholds of your graph. You'll see a fair amount of differences. And so what we've typically tried to do is instead, use various different ones and try to look for properties that seem consistent across each of those. And those are ones that we feel are more confident about in the end. So I guess I don't have one answer. I'll say that I've typically used spring embedding just because we have code for that most often, but it doesn't mean it's because it's a better layout system than others. Of course, I'm sure if you've dug into this, you'll know that some emphasize, different properties relative to others. And if your question has to do with those properties of how nodes that are spread further apart as opposed to clusters pushed close together, you may want to use a technique that's sort of emphasizes those metrics themselves. Great. And Kai, go ahead.
Hi, I just wonder if you also do some sort of structural connectons using diffusion MRI and kind of also combine it together with this functional connectivity fitting in some framework.
Yeah, I have not. So I don't do DTI work in my lab. I mostly do functional MRI work, although there's some really nice projects that are trying to do exactly what you're saying of sort of grouping the two together using different sort of modeling based methods of starting with the structural connectome that you might estimate from DTI or even neuroanatomical tracers and older work from say honey and sporns, and using that as sort of the skeleton and then placing the functional connectivity measures on top of those and trying to get an estimate of how could these functional connectivity measures sort of derive from this structural backbone. And I'm happy to sort of send references later, but it sounds like you're already a little bit familiar with that if you're interested, but it's not something that I personally have done in my own work.
Thank you.
Yeah, go ahead, Happy.
Hi, so my question was kind of related to what you were talking about coherence, in that finding regions that correlate in their frequency, almost like in one domain, how they change. And I was wondering how, it almost sounded very similar to MVPA, if I'm not incorrect. And I was wondering how those two actually differ in that sense.
Yeah, so I guess by MVPA, are you referring to like multivariate pattern analysis, sort of, you can sort of group data across multiple units?
Almost like RSA, yeah.
Yeah, so I think that, so MVPA or RSA are tools you could use with functional connectivity data and have been very effectively used. I won't talk about it today, but like say taking one of those matrices, you take the unique edges from that matrix and try to use that to say predict age or some other metric of interest by grouping information across the different functional edges. But they aren't per se like the same thing, right? Like MVPA can also be used on the activations from voxels of different regions, which would have nothing necessarily to do with functional connectivity.
But would those voxels be then categorized as being functionally connected?
Yeah, so not necessarily. So I think in like the classic Kriegs-Court sort of RSA analyses of like how people activations in response to say different images, those are that's a first order statistic of the evoked response in the bold signal to seeing that image. So it doesn't say anything about co-fluctuation between different brain regions per se. That said, regions that have common activations to the same stimuli over time are often also functionally connected. And so there's some older papers that show this across different tasks. There's even papers that have from, I think Ariel and Greenwald have shown this with neural signals that neurons in say V1 with similar orientation tuning also are correlated in their spontaneous activity patterns at rest. So you might think that there is a relationship, but a single like evoked response doesn't necessarily mean that two, even if they both have the same single evoked response, that the two are functionally correlated with one another. And the classic example that's often given for this is, you know, you might envision a scenario where someone is watching a movie with auditory and visual stimuli. And so in that case, you might have evoked responses from both primary auditory cortex and primary visual cortex, but they're not necessarily functionally connected with one another over time.
That makes sense. Thanks.
All right. I'm seeing some hands go up and down, so I think I've lost, but if anybody has any more questions, just feel free to jump in.
I have a quick question about the sampling rate, PR, how it affects the availability of the associated functional metrics. As you said, you know, having more samples may reduce the sampling error, but can also introduce systematic noise such as head motion physiology, you know, which in turn could bias the statistical estimates, right? If you have higher sampling rate, as in like more samples. So I was wondering, how do we select the right spatial and temporal resolution for resistance to MR?
Yeah, that's a great question. And again, it's probably not one that has a perfect answer. I will say that for fMRI, the information that you gain by going faster isn't that huge, just because we're also limited by the hemodynamic response in terms of how quick the actual bold signal moves itself. So most of the information we gain by pushing our TR to to faster and faster rates is more that we've gained the ability to separate out, say, respiratory effects or heart rate artifacts and so forth. And the trade-off there, of course, there's a trade-off with spatial resolution as well, so that you might have to think about it and other potential biases contaminating your signal, say by going to very high multiband factors. So I'll say in our lab, we see kind of similar results, whether we're getting a TR of 1 second or TR of 2 seconds or TR of 0.75 as you have in the HCP data set, at least at a general scale. Of course, that answer may be very different if you're in the EEG world or the MEG world, the ECOG world, where you may be interested in those other frequency bands. And so you may want to sample at a very different rate in order to say access gamma and so forth. So I don't think there's like one answer to that question across all of those domains, if that makes sense.
Thanks a lot. I have another quick question. So you elaborated on the usefulness of coherence and other frequency-based metrics, right? I was wondering, like, anyways, because of the slow hemodynamic response, there is not much. I mean, TAX is a low-pass filter, right? So there's not much frequency information available, and also we band-pass filter the response rate from MRI data. So how much what utility do we get by using a frequency-based analysis over, say, a time-based analysis?
Yeah, so I think the coherence analysis has been more commonly used in other domains because of that issue. I think in fMRI, I've most commonly not seen it to break apart different frequencies of the signal, just like you're saying, because we're already looking at fairly narrow band that we've bandpass filtered, but instead used as a way to separate out the coherence information, magnitude information from the phase information, which I think it still applies nicely to fMRI data. Does that answer your question?
Yeah, that does. Thanks a lot.
Okay, great. So I'm going to move along just in the interest of getting to some of these other examples at this point. But again, feel free to ask more of these questions. They're great. Keep them coming. Maybe they'll sort of intersperse with these other points as well. Okay, so in the next section, I just want to give you a few examples of how functional connectivity-based data has been used. And I'll sort of start off with a range of types of questions that people have been interested in asking with functional connectivity. And so these tools, this sort of ability to look at these functional systems in a living human, has inspired a lot of interest in whether or not we could use these tools to then ask, how do these systems respond when, say, a part of the brain is damaged? We've actually done some of this work with different lesions, for example, or if someone has a particular disease. You might also use these tools to then ask, how do these functional networks differ across people, and are those linked to individual traits? You might look at how these networks change or differ over the course of the lifespan with natural, developmental, and aging processes. And of course, you might also be interested, if you're coming from more a psychology, cognitive background, of how these different networks respond under different cognitive contexts or with different demands. And so I'll just note that these different types of questions do depend on these functional networks being able to measure signals at very different timescales. And so I'll come back to this point again in a little bit. And so one way, people have used various different paradigms to measure large-scale functional networks. And I really like this figure. This was from Thomas Nisolaris, Emily Allen, and Kendrick Kay recently had this nice review, if anyone's interested in, on the value of of dense sampling. And so let me explain what that means, or denser precision sampling. So most traditional fMRI studies, or many that are commonly conducted, usually contain data from maybe something like 20 to 30 subjects, each measured one time, so 1 hour of data perhaps. And recently there's been a move to push this in two different directions. So some studies have been shifting towards what you might call many subject, large and big data sets. where you have lots and lots of subjects, even pushing up into the tens of thousands now. And you have a small amount of data on each of them, and that can allow you to measure like population-based characteristics across them. So some common data sets of this sort are the Human Connectome Project, the UK Biobank, if you're interested in adolescents, the ABCD Project, and so forth. But recently, there's also been interest in pushing this in the opposite direction with functional connectivity data. And that is, instead of collecting little data on a lot of people, collecting only a small set of people, 1 to 10 individuals, say, but collecting lots of data on each of those individuals. And so, at least in the functional connectivity world, this movement was largely initiated by Russ Poldrack's My Connectome dataset. If you haven't heard of this dataset, it's it's publicly available. Russ Poldrack scanned himself twice a week for a year. So there's a lot of data, resting state data on him. He also has some task and other measurements as well. It's a great single person data set, get really detailed representation. And since that time, there's really been an explosion now of studies that are trying to do something similar, maybe in a slightly larger group of people or slightly different samples. And so my lab is focused most on this side of measurements, these sort of precision or deep measurements. We have a special issue that just came out if anyone's interested in reading more about it. Although sometimes we've used it in combination also, and I can talk about how you might combine with these sort of big data sets. And one plug I'll make, because I know you guys will be doing a hackathon, is that a lot of these data sets are publicly available. I'm giving you data today for your tutorial from the Midnight Scan Club data set, which I'll talk about in a second, but they might be really good data sets to start with for that. Okay, so as I said, one data set we've used a lot in my lab was a Midnight Scan Club data set. This is a data set that was collected at WashU just as I was arriving there for my postdoc. And so it was inspired by Russ Poldrack's data set, but instead of having just data from one person, they're like, let's have a few more. So they had 10 people, and each of these 10 people completed 10 functional MRI sessions. They also have a couple extra structural MRI sessions with different measures. And in each functional MRI session, the participants completed a resting state scan, as well as 3 tasks, depending how you count tasks, that were associated with motor, so like a motor mapping task, a semantic test where participants were asked to say whether a word was a noun or a verb, a dot coherence task, and then this was an incidental encoding task where participants were asked to categorize faces, scenes, and words that were repeated over time. And there were a few measurements that were collected outside the scanner on each of these participants as well. I will also note, you may note the unusual Midnight Scan name. So why is it called the Midnight Scan Club? It's because all of these data sets were actually collected at midnight, not because they were particularly interested in what's happening at midnight in these participants, but because the scanner was discounted 90% at midnight. So they were able to collect this data set at a fraction of the cost that it might be otherwise, which sort of gives you maybe inspiration for how you might go out there and collect your own data sets. At WashU, that's no longer possible because too many people were then scanning at midnight after this came out. So they increased the rates even at midnight, but maybe it's possible at other centers. Okay, so what can you do with these 10 subjects? So with this very large amount of data, you can start to make very detailed maps of brain networks in these individual participants. And so what I'm showing you here is the group average map you might have from the Midnight Scan Club. At the top, you may remember, if you happen to have very good memory, that this is actually very similar to the average I showed you earlier from 120 other individuals. So even with just 10, the averages start to look similar to one another. But beneath that, you can see the functional network map from each of the individual participants in this data set as well. And so you might notice that there are points of correspondence across all of these network maps. They all have roughly a default mode layout here in red. They all have like the visual system back here and so forth. But there are also points that seem different between each of these maps. And so you might start to ask questions about what do these differences mean? And I'll just point out that some of these differences are also things that some of these include elements that just are not present in the group average map. So for example, there was this little piece of the salience network here in black that appeared in many participants in this very rostral frontal region that isn't present in the group average map, probably because it moves around a fair amount and exactly where it's located across individuals. So in addition to looking at the layout, the spatial layout of these networks, you can also do other things with this large amount of data. One thing you can do is use resting state measures to parcellate the brain into different regions that are relatively homogeneous. And so what's shown here is the regional parcellation for subject 1 in the Midnight Scan Club dataset. You can also then use these techniques to map the complex systems of this particular individual in their own level and start to ask questions about how their brain network is sort of organized from a graph perspective. You can also use this large amount of data to test your particular measure of interest and its reliability over time. So because there are 5 hours of resting state data, you can take half of it aside and then use the other half and see how much data do you need to achieve a certain level of reliability. And so, for example, here you can see that for basic correlation measures of functional connectivity in the cortex, you probably need about 30-ish minutes of data in order to achieve high reliability on these measurements. And you can do lots of other cool things. For example, here you can look at the correspondence between these intrinsic resting state networks and task activation patterns in these data sets. This is so showing in the motor task, you can see the really nice correspondence between motor responses, activations, and the individually defined networks, which fit much better than, say, a group definition of where these networks occur. I also make a point that you can use this design, which has multiple subjects, sessions, and test states to look at how functional networks vary across different timescales or different contexts. So this is something that I worked on while I was in the lab. We looked at whether or not these functional networks can be thought of as relatively more trait-like aspects. You know, is it about the individual or state-like? Is it about what they're thinking about at a given moment in time, what they're doing? And so we looked at this in a variety of different ways. I'll just very quickly just show you one here, which is to say that what we compared the functional networks to one another. So in this matrix, any given cell is just a similarity between two of those correlation matrices I showed you earlier. So it's a second order matrix. And so it's organized first with subject along the main axis, within that by task, and then within that by session. And so when you do this, you see a number of different properties here. You'll notice that the background is not actually centered at 0, but around 0.5 or 0.6 correlation, indicating that there are commonalities across all these functional networks, like we saw in the previous graph. And then there's this big diagonal effect that pops out so that a particular person looks most like themselves, regardless of what day it is or what task they're doing. And so there are also other effects layered in here. There's a common effect of a given task, and there are individual by task and day-to-day variability factors. These were all significant in this data set, but you can see that they're substantially smaller in magnitude. So to us, we thought this indicates that you can conclude that functional network measurements are really more dominated by stable factors, intrinsic network organization that either changes very slowly over time or doesn't change very much over time, and with only much smaller contributions from state-based effects. Okay, so hopefully that gives you a picture of how you could use functional connectivity. And with that, I was going to turn to the last part of the talk and talk about the tutorial, but I'm happy again to take just a few minutes of questions before we jump into that. And I'll put up the tutorial information here while we're talking. Go ahead, Matilda.
Thank you, and thank you for this talk. I love your work, so thank you for being here with us. One question I have is in relation to this last point you made about stable effect over time, I would say. Do you have a sense of whether this is because, for example, these participants were healthy volunteers, kind of young, and whether this variation can be perhaps pushed by looking at all the participants or special populations?
Yeah, I think that's a great question and not one that we have data yet to speak to. I suspect that it'll be a similar general pattern that, you know, overall what we've tended to see across many contexts is there's a lot of stability in these networks. But I also, just like your question indicates, am quite interested, like in older adults, there's been this question, like, do they as effectively move across different task states? Maybe there's more variation. You might see that in certain psychiatric populations. So we're actually collecting some adult lifespan data in the lab currently with these sort of techniques to try to address some of those questions. And it would be awesome to see other people dig into that as well.
Thanks.
Yeah, go ahead, Hetty.
I had a very small technical question. If you can just go back to the brain image, one more. Yeah, right here.
more.
What's that big silver space?
Here?
Yes.
Yeah, so this is a cortical representation of the data. And so here is where, you know, all your subcortical structures would normally sit, which we don't have flat mapped.
That would make
So we tend to just zero them out. So you don't see them, and you'd represent those in a volume view instead.
Okay, thank you.
Yeah, no worries. All righty. So in the interest of time, since I know you all have other things to get to as well, I'm going to go ahead and start going over the tutorial. And so for this, I will open up the Python notebook in Jupyter Hub. I think I already opened it. I need to refresh it. Reshare my screen. Just going to share the whole desktop so we can kind of go back and forth on this. Okay, so hopefully you can get here with me as well. I think it's in your Jupyter Hub within curriculum and then connectivity. And so there's a bunch of files within here. The one you want to start with is called functional connectivity tutorial. It's the Python notebook. Although the other files may be relevant for you to look at as well. So feel free to sort of dig around as is useful for you. And so I'll just orient you first off that this tutorial has 4 basic components to it. And as I said, the goal of this tutorial was to give you a nice intro to a few very common and basic ways of looking at functional connectivity data that are useful at different levels of analysis. But hopefully, if you are already like an expert on these techniques, this is also just a good tool to start with and then sort of build your own like higher level things, maybe in the hackathon or whatnot, that you want to use later on in the week. So the overview is that it's going to use the midnight scan club data set that I just introduced. So you have some of the data already present there, both as time series and as well as some other metadata for one participant. And in the first part of the tutorial, I'll have you make what are called gray plots, which are good ways of looking at your original data quality, your time series, thinking about how you might denoise your time series. Then you'll make some correlation matrices and think about how you might interpret those and compare them. And then I'll introduce making graphs as well as hub measurements for these graphs. And so we'll see how far we get today in time, but I'll at least hopefully you'll have the tutorial to work through as well. And I'll try to at least briefly introduce each of these to you. And so let me just go ahead and start. I've also noted here that the Midnight Scan Club dataset, like the other ones I've talked about today, is publicly available. So it's on, I think this link is to OpenFMRI, but it's actually now on OpenNeuro. So I should have probably have updated that, but you should be able to Google for it. And the full data set is described in detail in this paper right here by Evan Gordon. So go ahead and get going. Just set up some things, some libraries that you need and some paths. And I incidentally also wrote this as just a like Python script originally, which is why some of the things are set up the way they are. So if you like using Python scripts, you can use this master NeuroHack Python script for it. And the libraries that it calls on for the more specific functions, don't know what's going on. The libraries that it calls on for the more specific functions are available, as I said, in here. So you can, again, feel free to go into these particular functions and look. Okay, so gray plots. What is a gray plot? Let's start with that. Maybe you are all familiar with it. There is a whole paper about these, sometimes called carpet plots or gray plots, that Jonathan Power wrote. It's something that I aspire to one day as well, have a single figure that I can write a whole paper on. And I find them very useful for thinking about data quality. Oops, excuse me. So I would encourage you to look at this paper if you're interested in digging in more deeply. I'll also note that if you are using fMRI prep for your analysis, they do spit out carpet plots for you. So that's a great resource as well. I'm including code that you can make your own, which I find useful because sometimes you'll want to make them slightly differently, right? Like you'll have different metrics that you want to look at or compare against. So now that you have the code, you can sort of go in and do that yourself. And so the output of this script is going to look something like this. And so The heart of this plot is this piece down here, which is the gray plot or the carpet plot, which is a bunch of different time series just stacked on top of one another. These are from the cortex, and you can have them from white matter and CSF below. And so the x-axis here is time from this particular session. And so typically when I've done these plots and in Jonathan's original representation, I actually fix also the scale, which I think can be quite important for thinking about the quality of the data, to only represent plus or minus 2% signal change, because if things are changing much more than that, that may not be a good sign for your data. You may want to be aware of it. Of course, you could make that more flexible within your data set, depending on what you want to do. So we usually mode 1000 normalize and then plot data with plus or minus 2% signal change. And so the other sort of things that you can play around with on this plot are the things that you put on top, which are usually some measure of artifacts or data quality that you might want to compare against. And so in the case of the ones that I'll have you plot, I have the original 6 motion parameters plotted here. And below that, I have 3 common metrics that are used for data quality that are placed in here. And so in red here, what you can see is the frame-wise displacement metric calculated in the power way. There are a couple different ways of calculating it, just as a note, which is just a measure of kind of grouping across all 6 of the motion metrics from one TR, one moment, one volume that you've collected to the next. How much has the head moved in total? And so you can see that many points are fairly low. I put a little line here of where we might usually mark a location as an artifact of having too high movement. We're pretty conservative about this. We'd say just 0.2 millimeters often for movement. And anything that's above that, we would say this is a bad frame if someone's moved. And you can see that, in fact, when there are spikes here, there often are also spikes in that gray matter time series below. So often when we're looking at these, we're trying to compare between the two and look for common signals that are present across all, at a given moment in time, across all of the gray matter voxels. So that's frame-wise displacement. The green line here is the global signal. So you might see that there are some shared aspects to these as well. And then the blue line here is a signal called d-bars, which has been used in some groups and not as maybe quite as commonly, which is a measure of how much from frame to frame the global time series are changing on average across the brain. So it changes a little bit more. Okay, so if we return to the actual data, to the actual code, if you were to run this, I've given you data from subject 1, and I've given it to you both right after the original pre-processing. This has just been, motion corrected, slice time corrected, and aligned. And then I've given it to you at each of the stages of what we call functional connectivity processing that we go along the way of denoising our data. So we first demean and detrend the data. We then do a regression of nuisance variables, which in this case includes white matter, CSF, global signal, their derivatives, and then the expansions of those. We then remove high motion time points and interpolate over that data. So that's what this step is. We bandpass filter the data, and then we demean and detrend the data again, just because I think this is a historic thing that's been left in here, but it's in your data set. And so you can make a plot for each of these steps, and that's what'll be what's spit out here at the bottom. You can notice at the end, there's nothing here because it hasn't been demeaned and detrended, so it doesn't fit within that range that I originally gave it. But once you've demeaned and detrended the data, you can look at what the original data looks like and start to look for patterns that look like artifacts or patterns that don't look like artifacts. We usually interpret things that are kind of present across the whole brain at once as not something that's really relevant for looking at spatially localized networks. And so you can see those signals, and it can be valuable to look at these both across different processing stages. So here you can see that once you do before and after doing these nuisance regression, you already get rid of a lot of that common signal across this data set. And then, sorry, go ahead.
Caterina, sorry to interrupt, especially because this has been a really great talk and I'm really anxious to look into this tutorial, but in the interest of time, we're kind of hoping to wrap up in the next 3 or 4 minutes. So just wanted to warn you. Okay,
good warning. So I will just give you the very brief overview because I know it's probably been a long day for many of you. So hopefully you'll be able to play around with this. You can compare across subjects, you can compare across different processing stages normally or runs. In addition to making the gray plots, I'll just sort of skip down. The next step will introduce you to the idea of how to make correlation matrix and the importance of ordering the correlation matrix. I think I saw a note very quickly in the in Slack that we didn't talk about parcellation. That's true. I did not talk about how you might parcellate the brain. I'm happy to sort of talk offline a little bit about that. And there's some links in the code about other references you might want to look up on that point. And you can compare across subjects and so forth. And then I introduced how to make these spring embedded plots with Gephi. They're actually force-based layouts, and how to color them based on different networks. So you can network metrics. So you can look at the degree, participation coefficient, or other measures. And if we were to open Gephi, which there are instructions, you would get something that kind of looks like this at the end for your networks. Or you can recolor these in this way. So you have it colored based on what network it is, or the degree for each participant, or the within module degree, or the participation coefficient. So sorry, I don't have time to go into it in too much more detail, but again, I am on the Slack channel. So if you, as you're going through, you have questions that you don't understand from the way things are written, just feel free to reach out and I'll try to respond as quickly as I can.